Okay, again, yeah, welcome everyone. So today's speaker is Dr. Larry Holler, and some of you may know him. I know Cindy had worked with Dr. Holler and Sue, and I know some of our faculty had worked with Larry before. So he's a professor in veterinary and biomedical sciences at SDSU, and Larry received his doctor of veterinary medicine from Kansas State University and his PhD from Washington State. So his main focus is on GM1 ganglioside, which is a brain lipid just showed has some therapeutic effects on neurodegenerative disorders, especially in Huntington disease. That's the one he's going to talk about today. He and his wife Susan, they founded the glycogen research. So the company's mission is to produce and promote the Ovine GM1 ganglioside as a therapeutic treatment for Huntington's disease. So with that, I'll turn it over to you, Larry. And I don't know if you're able to share the screen. You can share the screen, then you can take it from here.
Okay, let's host disabled participant screen sharing. That's probably not good.
Okay, you should be able to do.
There we go.
Okay.
All right, I won't be able to see you guys, so feel free to sleep. Because all I have up are all my slides. If you want to ask a question while I'm going along, it won't do you any good to wave your hands because I won't be able to see you, so feel free to interrupt. Title of the presentation is GM1 gangliosidosis, mad cows and Huntington's disease. And there's a bunch of sheep in the background. So if the title slide alone doesn't confuse you, let's hope during this next hour that I can sort of weave a story where all four of these things will kind of come together. Many years ago, probably 70 years ago today, researchers studying a family of genetic disorders termed lysosomal storage diseases made an observation on it's a really kind of a neat old technique of silver staining. These are actually Golgi stains of pyramidal neurons in the cortex of the brain. And they made the observation back then that can you guys see my little arrow? You can.
Yes.
Okay, so they made the observation that at the axon hillock region of the neuron, there was all these new neuronal processes. They weren't sure at the time whether they were axons or whether they were dendrites, but the knowledge at the time was that neurons, once they were developed, mature, could not develop new processes. So they all of a sudden kind of made the observation that something was causing these processes to be formed in these animals and humans suffering from lysosomal storage diseases, predominantly neuronal storage diseases. So these are what neurons look like if you just stain them. These are all the white fluffy material around the nucleus is all the lipid material, mostly lipid material that's accumulating in the cytoplasm. So combining the observation, they felt like there was probably something in this stored material that was triggering the development of these ectopic new either axons or dendrites. Basically, this was the sentinel observation that a company called Fidia, SPA, an Italian pharmaceutical company, took and made basically the hallmark of almost 75 years worth of research efforts, primarily into the field of GM1 ganglioside. Fidia, in their heyday back in the 80s and 90s, was a very going pharmaceutical concern. They had headquarters in Italy, Alberta Tremé, Italy, and Washington, D.C. They produced several products that were approved for pharmaceutical use in Europe. The first product I don't have listed was a product called Cronazole. And Cronazole was basically a preparation of total gangliosides. They had shown that in the stored material in these neurons was predominantly ganglioside, and they developed this product, Cronazole, as a treatment for peripheral neuropathies. As their program matured, they realized that the most active product in this total ganglioside preparation was GM1 ganglioside. And so they started an aggressive pharmaceutical development program, purifying brain gangliosides from slaughterhouse cow brain material. Fidia had dozens of patents covering everything about ganglioside production. The original GM1 molecule and probably a dozen patents on derivatives of GM1 ganglioside. Not the best GM1 picture, but basically what's important to notice is that you can see the sugar chains, the oligosaccharide chain attached to a sphingosine or ceramide molecule. The hallmark of the ceramide molecule is these long chain fatty acids. It's important to realize that GM1 ganglioside, we'd call it, it sounds like it's a single molecule, but based on the variation of the lengths of these fatty acid chains, the chemical composition is actually variable. So some of those chains will be 16 carbons, some will be 18, some of them have one double bond, others can have two double bonds. They occur predominantly in the outer leaflet of the cell membrane, plasma membrane. These sugars make this half of the molecule hydrophilic. The sphingosine side of the molecule makes it hydrophobic. So as you can picture, these long chain fatty acids insert into the cell membrane, and these sugar molecules stick out into the extracellular space and serve basically as the biologically active portion of this molecule. It's a natural molecule, so if you were to buy GM1 ganglioside on the market today, it will have been purified from most likely animal tissue. Our product we'll talk about a little bit later is a sheep GM1 ganglioside. You cannot synthesize GM1 ganglioside in the laboratory to any extent. Gangliosides in general are kind of an obscure field of science of biochemistry, and yet it's probably one of the more interesting and more important components of the cell membrane when it comes to a lot of different cell functions. I could give a whole talk on just cell functions with GM1 ganglioside, but we'll, for sake of time, its location in the cell membrane, it's involved in cell membrane integrity, in its binding interactions with cell membrane proteins involved with sodium and calcium transport across both ways of the membrane, turns on, turns off a lot of different nerve growth factors, and a variety of essentially important protein molecules that will relate directly to some of the neurodegenerative diseases we'll talk about. So Fidia in its heyday had human clinical trials going for spinal cord injury. It was one of the largest spinal cord injury clinical trials ever done. 750 patients enrolled. These Patients were usually treated for periods of 30 days up to some of them up to a year. Basically, the results showed that it improved the speed of recovery, but not necessarily the degree of recovery. The challenge with spinal cord injury is how do you get the treatment in to the patient in a timely fashion. So basically it became a challenge of recruiting patients, getting patients to trauma centers, deciding which category, the severity of their disease. It became essentially a data nightmare. Parkinson's disease, work done by Jay Schneider, some of the longest duration human clinical trials. He had patients that were on GM1 ganglioside for probably 5 years, some of them at very high doses. And basically the conclusion at the end of his clinical trial was that it actually seemed to put Parkinson's almost into, it slowed down the disease progression almost to the point of in some patients feeling like it was a remission. Stroke, a lot of patients enrolled in the stroke clinical trials. These, again, were a little difficult because you had to have a close proximity to a treatment center. No two stroke patients were difficult or were identical, so it was hard to categorize. A lot of stroke patients don't live past the first 2 or 3 days of the insult. The conclusion in stroke was that it actually did seem to improve the speed and degree of recovery, but it became a challenge, and the results were directly related to how quickly the patient was treated. Alzheimer's is probably one of the most interesting studies when you're looking at applications for GM1 ganglioside. We know in Alzheimer's that the beta amyloid, the toxic beta amyloid, forms and binds to GM1 ganglioside on the membrane. That appears to be the focus for the oligomerization of alpha beta amyloid and then the subsequent formation of the toxic component. Amazingly, the flip side of that coin is if you treat Alzheimer's patients with GM1 ganglioside that they had in a clinical trial done in Sweden, that at least the clinical parameters of the patients were greatly improved. Current research today suggests that both outcomes, that GM1 ganglioside is involved in the pathogenesis of Alzheimer's as well as a potential therapeutic, is possible, and it all depends on how much GM1 ganglioside is in the membrane, especially in the lipid rafts where the Alzheimer's, the A-beta protein forms. Coronary artery bypass was wasn't a lot of very big clinical trials. This is was kind of a unique little approach. Anybody that's ever had heart surgery will be put on a heart-lung bypass machine, and usually there is some damage to the brain. Obviously, it depends on a lot of other factors, but they basically showed that if you pre-treated with, and a lot of animal studies have shown, if you pre-treat with GM1 ganglioside, that you can prevent a lot of the subsequent post-surgical brain damage. So we've got lots of big diseases: Alzheimer's, Parkinson's, spinal cord injury. So what happened? Why didn't all this come to fruition? Right in the heyday of Fidia's GM1 ganglioside development program, mad cow disease broke out essentially it started in England and spread throughout Europe. Fidia's manufacturing process for GM1 ganglioside involved taking semi-trailer loads of cow brains from slaughterhouses all over Europe. And the obvious question became, how do you know that that tissue does not contain infectious prions? Within a matter of probably about 2 years of the development of mad cow disease, Fidia was essentially bankrupt. And pretty much their entire clinical program, with the exception of the Parkinson work, was terminated. So where does the story change? WH Land and Livestock, that's basically our agricultural entity. Glycoscience Research is the research and development entity. We realized we've been doing this for a lot of years. So when mad cow disease became prominent in Europe, and then the obvious problems that Fidia was having, we realized at the time there was a need to maybe find an alternative source to produce GM1 ganglioside. A raw material source, that could be somehow validated and traced. And that's what FDA was saying, is they wanted to know, you've got a semi-load of cow brains, they want to know where that third brain from the bottom came from. So Glycoscience Research and WH Land and Livestock basically started a development program utilizing sheep that had a lysosomal storage disease. So like I told you right from the very beginning, so sheep that are carriers for GM1 gangliosidosis, to explore using that model, that animal model, to produce GM1 ganglioside. So ovine GM1 ganglioside is a large animal model for the human disease, human GM1 gangliosidosis. It's an autosomal recessive genetic disease. Animals that are homozygous for this genetic condition accumulate GM1 ganglioside to 30 to 40% normal levels. That slide I showed you at the very beginning of all the swollen neurons, that's what the cells in these lambs' brain look like. Lambs are essentially normal at birth and progress to an end-stage disease by 5 to 6 months of age. Basically, these animals, the animals that are affected with GM1 gangliosidosis and humans affected with GM1 gangliosidosis are missing a single lysosomal hydrolase, beta-galactosidase. And so this beta-galactosidase is basically responsible for, taking and cleaving off this terminal galactose residue in the sugar chain. So the state of the art, I don't know if any of you have ever done any ganglioside or glycosphingolipid biochemistry, but these are TLC plates stained with resorcinol. Basically, the first 4 lanes are affected lambs. So this is a lamb in lane 1, this is how much ganglioside they are born with. So this is 1 day old. Lane 2, 120, 150, and then the last lane 4 is 180 days of age. Lanes 5 through 6 or 5 through 8 are essentially age-matched normal animals. So you can see, just looking at that large, essentially blob of GM1 ganglioside, is just how much ganglioside these animals accumulate. We knew that essentially we knew we had accumulation. We also knew that the observation made 70 years ago that the neurons would actually form brand new processes was taking place in these lands. This is actually a Golgi stain from the ovine GM1 gangliosidosis lands. So basically what we set out to do was to utilize a sheep GM1 ganglioside raw material source to produce GM1 ganglioside for pharmaceutical use. We developed a network of cooperator producers. We developed a PCR-based genetic test that could essentially, from a single blood sample, identify the homozygous affected and the homozygous normal from the heterozygous carrier animals. Developed essentially a processing facility and process in order to collect and store the tissues for GM1 production. And basically tissues are processed for by cryo grinding for storage. Over the years, and we've worked on this for pretty much over 2 decades, we've worked with researchers on spinal cord injury. I talked a little bit about some of the problems with that. How do you do clinical trials? How do you get close to everybody close to trauma centers? Classification of injuries. No 2 spinal cord injuries is identical. We worked for many years on Parkinson's disease. Parkinson's disease is actually, would be a tremendous application, but there was several problems with Parkinson's. One, you're talking 1 million+ patients with Parkinson's disease, and so the logistics of raising essentially the raw material to produce GM1 ganglioside would be challenging. The other challenge with Parkinson's disease was how best to deliver the product, how to deliver the ganglioside. All the clinical trials were done usually using primarily subcutaneous injections. The reality is that if you're trying to maximize delivery to the brain, diluting it out into the entire subcutaneous tissue is not necessarily very efficient. Intranasal is a more efficient method, and that's actually what we were working on when some of the basically when our collaboration on Parkinson's disease ended. They both were pretty challenging for clinical trial work. And then, lo and behold, a paper showed up in February of 2012 titled "Ganglioside GM1 Induces Phosphorylation of Mutant Huntington and Restores Normal Motor Behavior in Huntington Disease Mice." When this paper came out, it was almost too good to believe. To restore actual normal motor behavior in a devastating neurologic disease like Huntington's had never been reported before. So we felt like that was our new direction for clinical work. We had the fortunate essentially meeting with Dr. Stephen Hirsch. He's a remarkable researcher and physician at Mass General in Boston. He currently has moved into industry, working essentially still in Huntington's disease, but working for a pharmaceutical company. We worked with Dr. Hirsch for probably 7 years pursuing work in the Huntington's disease field. For those of you that aren't real familiar with Huntington's, it's an autosomal dominant genetic disease. So for me, it was always kind of unusual that we were trying to use one genetic disease to treat another genetic disease. I've always thought that was kind of unique. 30,000 people reported with clinical disease. 75,000 approximately have inherited the disease. So it's a dominant disease. So all they need is one allele of the mutant Huntington protein allele, and they will eventually develop the disease. 10 to 15 years preclinical. So basically they don't know anything is going, but the neuroimaging pathology has already started. 15, 20 years of motor cognitive behavioral decline. And then the last period of life is severe deterioration and dementia. Huntington's disease has often been referred to as one of the most terrible diseases out there. It's a combination of severe dementia and physical motor deterioration. It is a devastating disease. Even worse is the juvenile form of Huntington disease. This disease strikes children. The different stages progress very rapidly, much shorter course of period of disease. They may be diagnosed in their early teens. And the reality is most of the juvenile forms won't see 30. The length of the CAG repeat, which is kind of the hallmark in Huntington's disease, it's a series of CAG repeats on the end of the Huntington gene. The length of those CAG repeats correlates well with the severity of disease. The route of delivery, like any neurologic disease, neurodegenerative disease, is going to be critical in Huntington's disease. So these are some slides from neuroimaging studies that Dr. Hirsch had done, and it just basically shows the timeline from birth to death with the periods of prodrome stages, manifest disease stages, and then along the bottom, the temporal frontal cortex. So you see the increase in the yellow color. Basically what that is kind of a representation of the thinning of the temporal frontal cortex in the brain. And you can, it's actually starting, very early in life, but it's by the time by the time you're out towards the terminal end, it's a very devastating change. So the neurodegeneration associated with the accumulation of a mutant Huntington protein. So this is the protein with the CAG repeats. Interestingly, GM1 ganglioside has been shown to be deficient in cells, tissues of Huntington's patients. The actual mechanism of the deficiency is somewhat unknown, but they basically think the mutant Huntington protein is downregulated synthesis of all gangliosides. Restoration of GM1 ganglioside has been shown to drastically reduce the mutant Huntington protein that's accumulating by phosphorylating, basically phosphorylating the enzyme that's responsible for breaking down Huntington protein and prevents the accumulation. GM1 ganglioside, at the Ovine GM1 ganglioside actually was submitted to the FDA for pre-IND investigation and was reviewed favorably. So I don't want you to have to read this whole slide, but I wanted to highlight a couple things. In Huntington's disease, like in many neurodegenerative diseases, the current focus is on gene therapy, gene silencing. In this part here, in comparison, gene targeting strategies have a long and difficult development path because they require gene therapy or invasive approaches. Concerns about excessive lowering, so lowering the not being able to control how low you take the mutant Huntington protein, distribution of the treatment to the entire brain, off-target effects, and interference with the normal Huntington protein functions. We've argued with pharmaceutical companies that, while currently everything is being focused on gene silencing, GM1 ganglioside could be looked at as a therapy to fine tune the therapeutic approach. And then the bottom bullet, opportunity to profoundly improve the lives of families devastated by this horrible disease. This is Dr. Hirsch's words. That this is a man who has devoted his entire professional career to Huntington's disease, and it basically it's how important therapies are. Here's some work basically treating R62 mice. This is a transgenic Huntington's mice, basically with GM1 ganglioside. These are wild type mice. Looking at this is a FRET assay. According to Dr. Hirsch, it's an easy assay, but I've looked at it and I'm not sure I even understand completely how it's done. But basically wild type mice, levels of mutant Huntington protein treated with just basically sterile saline or artificial CSF, 30 mgs and 60 mgs of GM1 ganglioside. You look, the mutant Huntington protein levels in the 60 mg treated group is essentially identical to the wild type. Looking over here, this is basically phosphokinase B, some of the enzyme that they believe is responsible for phosphorylating and subsequently removing the mutant Huntington protein from the brain. While the difference wasn't statistically significant, there is a slight increase in the treated group in the AKT levels of phosphokinase B levels. So we think while it's not drastically different, some of these finely tuned regulatory systems don't need dramatic increases. This is some work actually it's actually still in progress. Dr. Hirsch, I'll kind of talk a little bit further. We basically wrote multiple proposals to NIH, an NCATS trend grant, which I think we actually would have been funded, except that the year we applied, they ended up not funding any grants. Dr. Hirsch, unfortunately, he's still kind of an advisor to us, but he's actually moved on from Mass General and into the private sector. But so now we work actually with a doctor. We'll kind of meet here a little bit. Dr. Gary Dunbar at Central Michigan University, and this is some data that actually fits pretty well with some of the work in Huntington's that's already been published using bovine GM1. So if you look at the red lines, those are Huntington's disease transgenic mice. These are the same mice as the R62s. The blue line are essentially the wild type mice. And then the green is the bovine GM1 treated, and then the black is sham treated with artificial CSF. So this is a test called the rotarod. You basically take HD, transgenic HD mice. It's a rolling rod. You see how long it takes them essentially to fall off. And so what you can see here is that it's very difficult actually to explain, but we're getting a pretty good treatment effect over wild type. And interestingly, it's a lot more pronounced treatment effect on rotarod than the bovine GM1. And we're not actually, to be honest, we're not sure why that would be. Chemically, they are slightly different, ovine and bovine GM1. But this last box down here is a clasping test. It's kind of almost reverse. So the score, the lower the score, the better they clasp, I think, is how this works. And so you see the ovine GM1 is performing substantially better than the bovine product. Hannah Hart this is actually Hannah's data, and I saw Hannah's on there, so she may be having to, there's a lot on here, so I won't really try to cover it all, but so these are basically Huntington's disease cell lines with multiple severities based on the number of CAG repeats they have after them. And so you got, these are the no HD, which I assume is a normal neuronal cell line. HD has 55 repeats, and the juvenile HD has 94 repeats. Interestingly, you're looking at cell survivability, an absorption assay. Kind of several things that struck me. One is the no HD mice no HD cell lines had a beneficial effect from being treated with GM1 ganglioside for survivability. And both the HD and the juvenile HD had treatment effects. It seems like it's a dose effect, and that up to a certain level, 0.2 micrograms per mils, 2 micrograms per mils, you saw a pretty strong effect, and then it decreases as the concentration goes up. It actually is a hallmark of GM1 ganglioside, and it always has been a challenge, is how to deliver the drug, and then what is the appropriate dose for essentially the application or area you're trying to treat. Many years ago when we started this, we kind of were under the assumption that, gee, this is a useful thing, and that treating devastating neurologic diseases should be useful, and that money won't be a problem. Lo and behold, money's always a problem, as you in research know. NIH, I'm not even sure how many NIH grants. We've actually submitted some NSF grants for manufacturing. Especially in a disease like Huntington's disease, what we have found is that the priorities within a disease like Huntington's is a rare it's a rare disease, an orphan disease. So you're talking a lot smaller population, and sometimes a lot easier approval through FDA. But like in Huntington's disease currently, gene silencing is king. And so any approach that essentially deviates from that focus will have substantial difficulty getting funded. And whether or not that's pressure from pharma, whether that's pressure from the Huntington's disease associations, if that pressure from who knows where, it's been phenomenally challenging for us to get public funding. HDSA, it's an organization, a large organization. The challenges is they basically are patient advocates, and they find themselves in a position where they can't advocate for funding for anything because the big pharma companies actually pay to run the organization. Nonprofits, we'll talk about a couple here really quickly. That's been basically our lifeline. Pharma, we've had lots of discussions with different pharmaceutical companies of a lot of different sizes over the years. I don't know for sure what is always the problem with the pharmaceutical industry. I think, to be honest, it's the natural source of GM1, the sheep. They don't, if they could take GM1, and several pharmaceutical companies have tried, if they could take GM1 and synthesize it in the laboratory, I'm confident that GM1 ganglioside would be currently available for Huntington's disease. We've had money from sheep organizations, because this is actually, for better or for worse, an unusual value-added agriculture project combined with a pharmaceutical development project. We've had some really generous private donations, people that, when they hear the story and they understand, or if they know somebody with Huntington's disease, have just decided they want to help. The Shepherd's Gift GM1 for HD is a nonprofit formed by Huntington's families. We're not on the board, we're not involved, but they have basically supported the development of GM1 ganglioside for Huntington's disease, and they continue to work. It's not an easy task. Bev Hardig Huntington's Disease Foundation. This is a private foundation in Indiana. Bev is I'm going to play you a short video. I don't know actually how old Bev is, but she's vibrant mother, husband, or wife, has a young son. She has Huntington's disease. Oops, hold on.
It's hard to hear.
Yeah, we can't hear that, Larry.
Oh, I'm not sure I can raise, hold up. Try one more time, see if it's. Can anybody hear that?
No.
Okay. Gary, so Dr. Gary Dunbar, he's currently work with Dr. Dunbar. Let's see if this short clip's a little louder.
No, unfortunately not.
Okay. So basically, I'll summarize. The Bev was basically telling how hard it is to, she's a young, vibrant mother and wife, and she has this devastating disease. And how do you deal with the progression? They channeled all their energy into forming this nonprofit. That video was shot in 2017. So we're 3 years later, and I'm pretty sure she's in a wheelchair now, or essentially in a wheelchair. Dr. Dunbar, he's a remarkable man. He's kind of a one-man show in Central Michigan. What makes him a very interesting collaborator is probably 30 years ago, he started his career working on GM1 ganglioside for traumatic brain injury, actually funded by Fidia. And so he kind of sought us out. His career started in GM1 ganglioside, and now he's probably will finish his career with GM1 ganglioside. The challenge is whether or not, whether we can generate enough data to pass the torch to keep the research going. Challenges for Ovine GM1 ganglioside treatment for neurologic diseases. The problems have, I think we've better understood the problems over the years, the multiple component parts. Something that starts with a GM, starts with producing a lamb that has a lysosomal storage disease, manufacturing of a natural product, and then pharmaceutical, development, all the hurdles and challenges involved in getting a drug through approval. Funding, we've talked a little bit about, NIH funding is phenomenally difficult. I think whether we like it or not, there's a substantial amount of politics involved, especially when you're in a small focused area like Huntington's disease. The funding is controlled by the organizations. It can be controlled by pharma. The nonprofits have kept us going. Venture capital, we've had lots of venture capital folks over the years court and talk. It seems like every time one shows up, you're in for about a year's worth of discussions. And what it always boils down to is that the pharmaceutical venture capital people don't like the agriculture component, and the agricultural venture capital people don't like the pharmaceutical development component. Big pharma, like I said earlier, if you could take and go to the lab bench and synthesize massive quantities of GM1 ganglioside, that you can control every step of production, that you didn't have a bunch of sheep producers involved, pharma would have already done this. Pharma has tried. They like the molecule. It's a fascinating molecule. There's some actually tremendous review papers on GM1 ganglioside if you just go to Medline and do some searching. Its story is not over. GM1 ganglioside is actually undergoing a phenomenal renaissance in China. China's producing GM1 ganglioside from pig brain. They don't care where it comes from. China's in the middle of an African swine fever outbreak, yet they're still producing gangliosides right and left. They just published a paper that came out in a Chinese journal, very clever approach using GM1 ganglioside as a treatment and a carrier molecule for an Alzheimer's treatment protocol they've developed. In animal models, it appears to be fairly impressive. The challenge for us in all the years we've worked on this is, we've gotten to know the Huntington's disease families, and that makes it phenomenally difficult. These patients and their families are, basically looking at a death sentence for the patient. It's an absolute brutal disease, and it, you get to know the families, and you want to try to help, but it's very difficult to navigate, the path. We've kind of sort of ran out of a lot of options. We keep surviving, and basically when funding runs out, I'm not sure what the next step will be. Questions? I'm back. Are there any questions? Oh, come on. Couldn't have been that clear.
Hello, Larry.
Hi,
This
Dr.
is
Tamala. How are you?
good to see you in our department. I've heard you before, but I have a different question from immunology perspective, because
Okay.
a lot of immune cells and epithelial cells also have GM1 gangliosides and other gangliosides.
Correct.
In gangliosidosis, in those conditions, are you aware of any immunological manifestations other than neurological?
I would suspect there are. We've not examined that. Immunologically, there's even in the live animal, there's unusual things that happen, and I'm not sure I could completely explain it. They actually tend to be in some ways protected against kind of boring bacterial diseases. You know, you don't see a lot of pneumonias in these lambs. We were actually going to do some immunology work, Alan Young, and we're going to do some flow cytometry and looking and see if, we could develop or detect any differences in lymphocyte populations. So, yeah, got a research project for you if you
Sure,
you want to do some work.
While you were giving, I was quickly looking for some of the things. I've noticed in Guillain-Barré syndrome, which is one of the side effects for some vaccines and things. So they do develop neuralgic pain and neuralgia, peripheral neuralgia. There are some reports that are connecting to these GM1 gangliosides to those. So I was just.
So the immunology story with GM1 ganglioside. So Fidia, back in their heyday, not only did they had to deal with mad cow disease, they had to deal with the perceived risk of Guillain-Barré syndrome. I don't know if you're familiar with Guillain-Barré, but it's basically a peripheral neuropathy, actually even a central neuropathy that back in the day, a lot of patients didn't survive. But it basically is an autoimmune disease where the patient is attacking his own predominantly myelin and a lot of central nervous system, peripheral nervous system tissue. And it turns out that one of the primary antigens in the nervous system being attacked is GM1 ganglioside. So for many years, everybody thought that, well, GM1 ganglioside injections would be causing this. And the reality is that no, bacterial mimicry is more likely the cause. Campylobacter jejuni is probably the primary offender, but even there's been some Guillain-Barré associated with COVID. So any of these infectious agents that have carbohydrate chains that can mimic GM1 ganglioside and trigger, the Guillain-Barré is probably the most famous, but there's a whole family of them.
Right. A lot of bacteria do have lipopolysaccharides with polysaccharides similar to, yes. Thank you.
It took Fidia years to overcome that stigma. Actually, their product Cygen was pulled off the market in Europe, and at the time was probably one of the 5 most prescribed drugs in Europe. And it was pulled off the market.
So is there a dye
basically very simple epidemiology showed that it didn't make sense, because there was the incidence of Guillain-Barré was the same or higher in countries that had never used ganglioside.
So do you have any dye that stains the polysaccharides on your GM1 ganglioside?
Dyes that'll stain, well, I mean, any kind of carbohydrate stain will light the cell up. They're all non-specific carbohydrate stains, but yeah, you can light them up. Yeah, there are some antibody reagents. The reality is it's phenomenally difficult to make an antibody to GM1 ganglioside or to any of the gangliosides. They're just not immunogenic. And so you have to attach a ganglioside onto some great big ugly carrier protein in order to get an antibody. And I remember historically the antibodies that were being sold commercially were terrible. You know, we'd try to do some immunoblotting and you could not get anything to stain. So,
Thanks, Larry.
any other questions?
Yeah, so Larry, it's an interesting work. So you talked about the delivery. I mean, I was just wondering, how does it get into the brain? Is it what kind of vehicle they use to cross the blood-brain barrier?
So it's been probably one of the more challenging areas. GM1 ganglioside actually has some active transport mechanisms across the blood-brain barrier. So it can get there. I mean, we know from a lot of Fidia's original early work that even injected subcutaneous, that you can get GM1 ganglioside, you know, whether you're radio labeling it or whatever, you can get it into the brain. The problem is it's a fraction of what you injected. So it's not particularly an efficient mechanism. With Huntington's disease and all the gene silencing, all of those approaches are going to be delivered intraventricular. So, you know, we thought, and we still think, and actually the Alzheimer's work that I talked a little bit about, that was all done with a catheter and an implanted pump and slow. I think the Alzheimer's work was 30 milligrams a day, you know, for a period of a year. And so basically, the more direct the delivery, the better the treatment approach will be. Subcutaneous, you know, that's all the Parkinson's work was done subcutaneous. Dr. Schneider also did some experimental stuff that most of it never got published, but on intranasal delivery. And he actually felt that was by far the preferred method, especially for the early or the asymptomatic and or early. And we kind of felt that even for Huntington's, that made sense. So if you're a asymptomatic Huntington's patients, take an intranasal, you can get the ganglioside across the blood-brain barrier at the olfactory epithelium. You know, if you're symptomatic already, then, you know, maybe a more aggressive delivery would be warranted. But the fatty acid chains tend to be the sticking point for getting stuff across the blood-brain barrier. They've shown actually recent work, and it's kind of a neat approach. They've shown if you take just the oligosaccharides, so cleave it off from the lipid, that you can actually obtain most of the pharmacologic activity and in animal models and cells, whatever you're using it, without the lipid. You know, so anything that involves just the oligosaccharide portion binding, you know, so if it's going to bind to whatever effector protein, you know, whether it's the phosphorylase or whatever, that you don't need the big lipid. Now, the part that I kind of chuckle about, in order to produce their oligosaccharides, though, they have to start with the whole molecule and cut the lipid off. But, you know, if it works better, I'm okay with that. You know, that's I know most of the people doing that. I was, you know, said, well, if it will get it to where it across the blood-brain barrier, get it to the tissue it needs to, that's, you know, yeah, you can. I've got a lot of GM1 ganglioside. We can cut it in half.
So other questions for Dr. Haller?
Yeah. Hi, Larry. I have a question.
Hi, Cindy.
So what are the challenges with synthesizing GM1?
So it's the biggest challenges are playing with the sugars, getting the sugars to attach. So you can start off the lipid backbone, the sphingosine, the ceramide molecule. It's getting the sugars to go where they're supposed to go without attaching. So you're just basically ugly organic chemistry. You add one sugar and then you block it, and then you put another one on, and you know, there's been people trying to clone or get an E. coli to produce the carbohydrate. Well, yeah, it can. Problem with E. coli is it doesn't know just to produce the one you want. And so you're forced with trying to separate out your particular, the oligo from GM1 out of the mess of what all the bacteria produced. The Chinese have been successful in actually doing some total chemical synthesis. And I mean, we're talking page after page after page after page of reactions. And, at the end of the day, you can see some guy in a laboratory throwing his hands up in the air saying, I've been successful. But he might have manufactured, 0.01 micrograms. So, when you're looking at a pharmaceutical use, you're talking kilogram quantities. So, I mean, it's the carbohydrates that give you the headache.
Okay, so in the interest of the time, so we'll stop here. So let's virtually thank Dr. Holler.
Thank you.
So thank you, Dr. Holler. So I'll send you the recording. So again, thanks to you and Susan for presenting to the department. Thank you very much.
Thank you.